All right, so let's talk about the urinary system and our acid-base balance lecture. Our urinary system is a system with not very many organs. We're really going to kind of focus in on 6 total organs. Your 2 kidneys, the right and left, which is where the majority of stuff takes place, where we filter our blood, where we make urine, we return vital substances back to the blood. The urine from there is going to drain down by way of 2 conduits called ureters to our holding tank, our bladder, which holds it until it's time to be peed out. And then we use the urethra to get it out of the body. Now, looking at this figure, this would be of a female, and there's a couple different things to kind of give you that idea. But the main reason why I say that, easiest thing is you can see ovaries and a uterus, which men do not have. A big difference is the location of some of these organs in respect to one another between the men and women. But a big thing is that the urethra in females is a entirely separate structure from her reproductive system, whereas the urethra in men is something that we'll talk about in the next lecture as well, because it's a shared tube. It not only carries urine for urination, but it's also a conduit that carries our semen when a male ejaculates. So it's a shared tube between our urinary and reproductive systems. We'll get into all of that here later in the lecture. For the most part, what we're going to spend our time in this lecture talking about are the 2 kidneys. The right and left kidney are where, like I said, most of the magic happens. And this is really where most of the functions of our urinary system come from. All right, let me give you guys a second or two here to write. And you might need to chime in if I go too fast or whatever. So if anybody gets behind, just holler. Okay, once again, the 6 organs of the urinary system include 2 kidneys, 2 ureters, the bladder, and the urethra. Kidneys are described as the primary organs of the urinary system, and they are what provide us with these different functions. First of which, regulation of blood ionic composition. Remember, ions are things like sodium, potassium, calcium chloride, hydrogen phosphate. They're all listed there. We need to maintain perfect concentrations and compositions of those in our blood, and our kidneys are responsible for doing so. And our first slide there on the previous page, we said that we're going to be talking about acid-base balance. Thinking back to the very beginning of the semester, I told you there were 2 main systems that help regulate our acid-base balance, or our pH. We very recently talked about the respiratory system. By controlling how much carbon dioxide we breathe out, we control our blood pH. But for more long-term acid-base balance, our kidneys are involved. So part of the urinary system, by excreting out variable amounts of hydrogen ions and conserving bicarbonate ions, we can maintain our blood hydrogen ion concentration at an adequate level. Thinking back, we've talked about the term blood volume before. Blood volume refers to the amount of blood that we have. And as you guys know, I've made sure I hammered it into your brains. Around half of our blood is just water. So by conserving or eliminating water in our urine, we can change our blood volume. Okay? And as we talked about here not too long ago, any increase in blood volume increases blood pressure, and a decrease in blood volume will decrease blood pressure. BP, short for blood pressure, is a thing that we can regulate using our kidneys to regulate blood volume. But on top of that, our kidneys secrete an enzyme we've talked about before, renin, that activates that renin-angiotensin-aldosterone pathway that can lead to increases in blood pressure. So it regulates blood volume, which regulates blood pressure, but it also just directly regulates blood pressure by releasing that chemical called renin. It maintains our blood osmolarity. So separately, it regulates water and loss of solutes in our urine to help maintain osmolarity, which osmolarity is a measure of our total number of dissolved particles per liter of solution. So essentially, it is a measure of how much water compared to other stuff is in our blood. So by changing water levels, essentially you change this measure of dissolved particles per liter of solution. So you can kind of think of osmolarity as just kind of a simple, or in simple terms, like just water balance is kind of how I tell students to think of it. Any change in osmolarity generally reflects changes in water in that solution. So kind of straightforward there. Two hormones are produced by our kidneys. Calcitriol, which we learned about, helps regulate blood calcium levels. We release this when our calcium levels are low. We talked about that back in our endocrine system lecture. And erythropoietin, which we talked about back in our blood cell lecture, because we learned that it makes or it stimulates our red blood cell production process, what we call erythropoiesis. Our kidneys help regulate our blood glucose levels, blood sugar levels, by aiding in gluconeogenesis, which is one of those 6 buzzwords that I had you guys know at several different points this semester. Remember, gluconeogenesis is the formation of glucose from non-carbohydrate sources, from things like, in this case, the amino acid glutamine. And our kidneys can also release glucose in our blood to help maintain blood glucose levels. We don't release a lot of it, at least in a normal, healthy individual. One of the telltale signs of a person that has diabetes is urine that has way too much glucose in it. That's not something that should normally be happening in high numbers. And then excretion of wastes. Wastes that are formed from metabolic reactions, as well as foreign substances, which could be drugs or toxins. And as you can see, there are a lot of different body wastes that we need to get out in our urine: ammonia, urea, bilirubin, creatinine, uric acid, drugs, environmental toxins. Let's talk a little bit more about those here on this page. Okay, just defining what a waste is. It's just any substance either useless to the body or present in excess of our body's needs. So either we have no use for it or we just have too much of it. Kind of a subset of those would be our metabolic wastes, which are waste substances that are produced by our body when we're going through metabolism. So, thinking back to our last lecture, hopefully everybody's had an opportunity to watch the rest of that lecture. We talked about, how we make ammonia when we break down our proteins, specifically the amino acids in our proteins. We learned that ammonia is converted to urea, which is much less toxic to our body and is, and as I said in that lecture, a very strong component of our urine. Both ammonia and urea will be classified as nitrogenous wastes, which are toxic metabolic wastes, and pretty straightforward. Nitrogenous wastes contain nitrogen. Okay? So urea, one of those, byproduct of the protein catabolism, breakdown of proteins produced by our liver when we convert it from ammonia. Uric acid is a byproduct from the breakdown of nucleic acids. So breaking down DNA and RNA will form uric acid, something that we must get out of our body. You ever heard of gout before? Gout is a condition in which people have excess uric acid in their blood and not getting it out. It's very painful, especially it affects the joints for whatever reason, and it's not a pleasant condition for anybody to suffer from. And then creatinine, you're probably thinking right off the bat, just based on its name, about creatine. Creatinine is the byproduct that's produced when we break down our creatine phosphate, which, if you remember back in our muscles lecture, we learned that creatine phosphate was that ATP resource that we use in high intensity, really short duration exercise type activities. So, all right, we can measure our levels of nitrogenous waste in our blood using what we call a BUN, blood urea nitrogen test, which is, the name is exactly what it is, measuring the urea nitrogen in your blood. Condition called azotemia is an abnormally elevated BUN and may indicate renal insufficiency. Now, anytime you see the word renal, renal is a direct indicator that we're talking about the kidneys. And insufficiency just essentially means that the kidneys are not functioning adequately. A person that suffers from azotemia a lot of times is going to have to undergo hemodialysis, or as a lot of people just call it dialysis, where basically they use a machine that filters a blood form like their kidneys should be doing. It's a pretty how do I want to say this? Pretty taxing process for a person to have to do this. They can't just do it once and then be good. If they're having kidney failure, they're going to have to do this, every couple of days. Otherwise, they're not going to, survive very long. And it's a pretty long process where it takes several hours. And what they have to do is have basically two lines you can see here into an artery and a vein. One draws the blood out of the body into the machine that filters out the blood, not removing the blood from their body, but removing the waste from their body, putting in some kind of new fluid. And then along with that fluid, the blood returns back by way of the other line. So it takes like several hours. And people that suffer from kidney disease, kidney failure, really, their whole life revolves around making sure that they can, get to a place that has a dialysis center and can, perform their filtering. Otherwise, their body's going to become toxic and they're not going to survive very far or very long, I should say. So, not a fun experience for anybody that has to go through dialysis. Okay, on to talking about the kidney structures known as nephrons. Within each kidney, there are about 1.2 million of these little functional units called nephrons, each of which consists of two principal parts, what we call a renal corpuscle, which is where we first filter stuff out of our blood and form what's called filtrate, which is just a precursor to urine. And blood comes in by way of a set of blood vessels into this kind of blobby-like structure. We pull out a lot of water and a lot of other stuff from the blood. And then we run it through this long winding, what we call renal tubule, which there's several parts to it: a proximal convoluted tubule, a loop of Henle, a distal convoluted tubule, collecting duct. And then what we end up doing is just kind of passing stuff back and forth between the tubule and our blood vessels that surround them. Ultimately, by the time we reach this point, we've essentially made the urine that will then filter out of the kidneys and then run down to the bladder by way of those ureters. But it's a pretty complicated process. And I say pretty, I should really kind of warn you, it's very complicated. So let's talk about kind of the anatomical features of, in this case, our renal corpuscle first, and then we'll get on into the renal tubule. That figure that was on that previous page is a fairly simplistic version of a nephron. Here's a little bit more detailed picture of it. As you can see, we've added blood vessels kind of all over the place. You can see they surround the renal tubule all over the place, which is a great way for stuff to get back into the blood or to leave the blood and enter the tubule. So I'll kind of bounce back and forth with this picture where we can kind of see the parts and these next couple slides here. Looking at this figure here, essentially what we're doing is just kind of zooming in on this segment of the nephron at what we call its renal corpuscle, which, as I said, is where we first filter our blood and form what's called filtrate, or it's more specifically called glomerular filtrate. Each renal corpuscle consists of kind of three major types of blood vessels: an afferent arteriole, which is the vessel that brings blood into the corpuscle, massive capillaries called a glomerulus, and then another arteriole. This is called an efferent arteriole that allows the blood to leave that massive capillaries. So kind of going through, we'll start with talking about the afferent arteriole. This is a very distant branch from the renal arteries that bring the blood into the kidneys in the first place. They come in, and through a series of branching, eventually we form these afferent arterioles that bring the blood in to the corpuscle, okay, inside of the capsule. The capsule is known as a Bowman's capsule. The capillaries here, you can kind of barely see them, are the little red parts to our picture right in here, but you don't see them very well because there are these cells that are surrounding those capillaries. Okay, those cells are called podocytes, okay, which look always to be like little bugs. Okay, little bugs with lots of little legs. Each of those little legs is what we call a pedicel. Okay, so let me just kind of read through, and I'll kind of get to all that. So afferent arterioles bring blood into the renal corpuscle, where it turns into a massive capillaries called glomerulus. The function of the glomerulus is possible due to a large pressure gradient. Blood in those glomerular capillaries is under high pressure due to having branched from the aorta. So substances in the blood that are small enough to pass through the pores in their capillary walls are forced out into the Bowman's capsule. So there's a lot of pressure inside of these blood vessels. It's going to try to force water and small substances out into this Bowman's space. Okay, and then it's going to filter out into what we call the proximal tubule, which is the beginning of that renal tubule that I was just mentioning on the previous page. Now, the entire structure that surrounds the glomerulus is called a Bowman's or glomerular capsule. It is two-layered capsule encircling the glomerulus. Kind of the two, the least important is this parietal layer, which is kind of just encloses the whole thing, so makes it watertight. The other layer is the visceral layer made up of those podocytes that I was talking about. Okay, so reading through, glomerular capsule is composed of an outside layer of simple squamous epithelium, inner layer of modified simple squamous epithelial cells called podocytes, and those are going to wrap around the capillaries. Okay. The little finger-like or leg-like extensions coming off of them are called pedicels, okay, and they mesh with adjacent podocytes, forming a dense network around the glomerular capillaries. Okay. So essentially what they do is they wrap around the capillaries, and they're like a coffee filter, and that they're going to wrap around the blood capillaries, but they're not going to prevent water and really small substances from leaving the blood and entering that space. Now, they will help prevent big things from leaving the blood, like our blood cells and our plasma proteins. But anything that's super small is going to be able to pass through that set of layer or those sets of cells fairly easily. Okay. There's the little spaces in between the cells, so that's where you can see the little red. That's what we call filtration slits to allow the filtrate to enter the capsule. Okay. Glomerular filtrate is the fluid that collects in that space. Usually I just call it filtrate, but it is formed from the glomerular capillaries and then exits into the renal tubule, specifically what we call the proximal convoluted tubule or proximal tubule as it's listed here. And then what's not taken out of the blood exits the capsule as what we call an efferent arteriole. From our renal capsule, the filtrate is going to run through a long, winding, hollow tubule called a renal tubule. And there are several parts to a renal tubule. Overall function of the renal tubule is to collect the urine and concentrate it by exchanging stuff back and forth with our blood and kind of just providing fine control of what we end up releasing out of our body as urine and return back to the blood. Begins with what we call the proximal convoluted tubule, which on the previous page, which is called the proximal tubule. The word convoluted is just kind of a fancy word for like a jumbled up. mess. And you can kind of see why we call it a convoluted tubule. There's actually a proximal and a distal convoluted tubule. Proximal convoluted tubule is where that filtrate first goes after it leaves that corpuscle. So it's going to be the water that was able to escape the blood along with the other small substances. And this is where we actually return a lot of that stuff right back to the blood. So it's kind of crazy, but we pull out a lot of water and a lot of substances from our blood here, and then we just give it right back here. Sounds really stupid, but that's what we do. And notice there's a lot of red squiggly lines surrounding that proximal convoluted tubule and the rest of the tubule. This is the route in which blood is able to pick up stuff from inside of the tubule, performing what we call reabsorption. And we're also going to be putting stuff from the blood into the tubule, kind of like what we did here, but we call that secretion. Okay. Now, the proximal convoluted tubule is made up of a layer of cuboidal epithelium. So the cells are cuboidal with lots of mitochondria and a prominent microvillus or brush border that faces the lumen. If I go back to this previous page, we can see that right here. And so the cells is a single layer, so it's simple squamous, or simple cuboidal, cube-shaped cells. There's where the cuboidal comes in. And then notice they've got the little microvilli forming what we call its brush border, just like what we talked about in our small intestine within the villi of our small intestine. We have cells that have little finger-like extensions, increasing their surface area. It's going to allow them to pull stuff out of the tubule and return it to the blood vessels that are surrounding them. So there'll be blood vessels all around it. And then also to allow stuff to come from the blood through them and into the tubule. So like I said, we pull a lot of stuff out of the blood, and then pretty much immediately we give it right back here in that proximal convoluted tubule. And so the brush border and the access to a large energy yield, because of its numerous mitochondria, allows for a great deal of what we call reabsorption and secretion to occur. We'll talk about what reabsorption and secretion are here in a little bit. Key feature of the proximal convoluted tubule is it's contained strictly out into what we call the renal cortex, which is the very thin outer shell of our kidneys. And whereas the next section of our renal tubule dips down kind of more into the interior of the kidney into what we call its renal medulla. Now that second segment of a kidney, or excuse me, of a nephron is called its loop of Henle. Loop of Henle is also known as a nephron loop. So the same thing, it's a kind of U-shaped segment to the nephron where we have a descending limb that dips down into the medulla, and then we take a 180 and then ascending limb that goes right back out into the cortex. Okay. Now, the cells of the loop of Henle can vary from simple squamous epithelium, which means you've got a single layer. Squamous tells you that they're flat-like cells. So thinking like what we talked about with the alveoli, the type 1 cells in our lungs that help us bring oxygen into our blood, we're simple squamous. Single layer, very flat plate-like cells, so stuff can easily pass through them. They're simple cuboidal, so kind of like what we just saw with the proximal convoluted tubule. Simple one layer, cube-shaped cells. And then there's also simple columnar, which means that they're just kind of taller shaped cells. So they're a column shape, but they're still only one layer thick. The cells in this loop almost entirely lack microvilli, so they don't have those little finger-like extensions that we saw back in our PCT. Now, I mentioned in that figure what we call the descending limb and the ascending limb. The descending limb descends down into the medulla. It contains what we call a thin segment that suddenly narrows as it descends, whereas the ascending limb is the section of our loop that turns 180 degrees, returning back to the cortex, and contains a thick segment that widens the tube as it passes upward. So looking here in our illustration, it's kind of hard to kind of really see it without me pointing it out to you. But just look at the width across in this area in our ascending limb. versus a crossover here in our descending limb. Can you see how much skinnier this is over here than it is over here? It's not very drastic, but we would call this segment of the loop of Henle a thick segment and this a thin segment. Now that's important, not for appearances, but for function. The thick segment is made up of simple cuboidal epithelium, and the cells are engaged in active transport of salts and therefore need extra mitochondria for that ATP production, because we always know active transport requires ATP. That makes the cells a little bit thicker. Okay. The thin segment is thin, which is important because it makes it very permeable to water. Water is going to be easily able to pass in and out of the descending limb. It's not going to be able to move in and out of the ascending limb. But what the ascending limb does is it's going to be pumping salt out into this renal medulla. They say actually if you were to taste the middle part of a kidney, which most people in America don't really eat kidneys, but this area of a kidney is going to taste really salty, which is because of what's taking place here. And then, as we talked about in our digestive lecture, wherever salt is, water is going to go with it. So the movement of salt out of the ascending limb into that renal medulla is going to actually be what draws the water out of the descending limb. So it's kind of a crazy thing, but there's an interaction of stuff going on here that affects the movement of water. From the proximal convoluted tubule, or excuse me, from the loop of Henle, our tubule goes back up and out into the renal cortex. And we jumble back up and form what's called a distal convoluted tubule. The distal convoluted tubule, or DCT, is strictly out in the renal cortex, so it's contained to the renal cortex. And its job is to conduct filtrate out of the nephron and into the last segment, what we call a collecting duct. And now looking right here, well, there is a segment of that distal convoluted tubule that happens to pass right by that afferent arteriole. Right here, we call this a juxtaglomerular apparatus. Okay. And the word juxtapose is a term like if you like our photography student, or you take pictures or something like that. Juxtapose just means like next to. So the juxtaglomerular apparatus is this segment of the renal tubule that passes right next to that afferent arteriole and that glomerulus. And there's actually communication that exists between these cells here and the cells here. If I go back to this picture, I didn't talk about this little yellow oval-shaped area, okay? But this is that distal convoluted tubule as it passes, and it's literally touching that afferent arteriole. Okay? The cells that face the afferent arteriole are called macula densa, and they can communicate with these juxtaglomerular cells of the afferent arteriole. Okay? So there's communication that can go on here, which is going to have a drastic effect on the beginning processes of the making of the urine. Okay? So let me just read through. Juxtaglomerular apparatus, the portion of the distal convoluted tubule, pushes past the afferent arteriole. This interaction helps maintain homeostasis of blood flow. Granular juxtaglomerular cells are specialized muscle cells of the afferent arteriole. So that would be what these are here. They contain mechanoreceptors that monitor pressure of the blood in our afferent arteriole. The macula densa are specialized cells in the adjacent distal convoluted tubule, and they contain chemoreceptors that monitor the concentration of sodium and chloride ions in the urine inside the tubule. Now, essentially what is going to take place is as we filter out blood and form filtrate, that then is going to trickle all the way through our different segments of the tubule. Let's say we're pulling out way too much water, and we're going to end up peeing out way too much water. And in doing so, we're also going to be losing valuable ions and other stuff from our blood. So if we're like having a big rush of fluid through the filtrate and it's running really, really rapidly to the point where we're going to lose those substances, the fact that we communicate right here between the tubule and the afferent arteriole where everything began, what we can do is send a signal across from here to decrease the size of this afferent arteriole, which means less blood's going to come in and it's going to slow it down. Okay. I always use this as like an analogy of working in a factory as part of a a conveyor belt team where you're like putting together a doll or a Barbie doll or some type of toy, where if the conveyor belt's moving way too fast, everything's happening too quickly, parts to the doll are not going to get put on the Barbie doll, and at Christmas time the little girl wants to get a Barbie doll without a head or something like that. So it allows for a person down the line, or in this case the DCT, to say, hey, hold up, we're moving way too fast, so it can slow down the conveyor belt. And we'll get to talk about all that here in a little bit more in just a little bit. And from there, the last segment of our nephron is what we call a collecting duct. Okay, it's called a collecting duct because at this point we can essentially almost call it urine. By the time we get down to this point of the collecting duct, it is urine. Now up here, we're still doing a little bit of the finalizing of that fluid. And then by the time it gets down here, we've done everything we really can do to it. So we'll stop calling it filtrate by this point and call it urine. Now notice here in our figure, we've got these little branches coming off of it that are going nowhere. It's not really the way that they are. A collecting duct is actually a shared duct with multiple nephrons, meaning that like this little branch here that's going kind of nowhere, well, there will be all of this attached to it, over here, and another one over here, another one over there. So a collecting duct collects that filtrate and ultimately urine from multiple different nephrons. Okay. Now, like I said, towards the end of the collecting duct, we can essentially call it urine by that point. But there are two cells at the very end of the distal convoluted tubule in the beginning of the collecting duct that can do a little bit of last second altering of that filtrate, either to pull stuff from the blood and put it into the filtrate or to remove it from the filtrate and put it back into the blood. We have what we call principal cells that respond to hormones aldosterone and antidiuretic hormone. And both of those are going to end up kind of pulling water and salt out of the filtrate, returning them to the blood. And then intercalated cells that secrete hydrogen ions. And that means that they're going to be going into the tubule, and meaning that we're going to be peeing out hydrogen ions, which is going to be helping to control our pH. Okay, so that is the kind of just anatomical overview of a nephron. Very complicated little parts to our kidneys. They're microscopic in size, but they're obviously super, super important. And we've got over 1 million of them in each of our kidneys. Now, I've been talking about the production of urine here as I've kind of run through the anatomy of things. I've used these three terms, maybe just not the full term. I've used reabsorption, I've used secretion. Those are short for tubular reabsorption, tubular secretion. But before you get to that point, you have to perform glomerular filtration, which I talked all about on that slide where we looked at the anatomy of a renal corpuscle. This illustration here at the bottom of the page, just a very simplified version of this, and I'm talking super simplified, where we're first pulling stuff out of the blood into the glomerular capsule. We're performing what we call glomerular filtration, the very first step of urine production process, where water and most solutes in our blood plasma move across the wall of the glomerular capillaries, where they're filtered and moved into the capsule. And then from there, we run it through that long winding renal tubule. Throughout the renal tubule, we're going to be returning stuff to the blood through what's called tubular reabsorption. Okay, so this is going to occur as filtered fluid flows through the renal tubules and collecting ducts. Tubule cells reabsorb about 99% of our filtered water. So like I said, we pull out a lot of water, but right away we give most of it back. About 99% of that water that we pulled out gets brought back into the blood. So it seems really stupid, but that's just what we do. And along with that water come a lot of many useful solutes that we, need to keep in our body and don't want to lose in our urine. Now, we've learned about reabsorption before. Back in our lecture of or on blood vessels, we learned that reabsorption refers to the return of substances into the bloodstream. We recently talked about digestion, and in that lecture, we talked about absorption, which is the movement of stuff like through the walls of our small intestines into the blood. Okay, so like thinking about like water, when it's brought into our body, when we drink it, we pull it into our bloodstream through our walls of the GI tract. That's what absorption is. Well, that water that we bring in eventually is going to be filtered out and made into filtrate, but we're going to pull 99% of it back into our blood. So we're reabsorbing. And when you add the letters RE at the beginning of a word, like redo, you're doing it a second time or a third time. Okay, you're doing it again. That's what reabsorption is. So just absorbing, returning stuff to the blood, after we've done it already before. The opposite of reabsorption is tubular secretion, which is the movement of certain materials like wastes that we talked about earlier, drugs, excess ions, into the tubular fluid. And I really like the fact that they use an arrow that's colored yellow because, urine a lot of times is nice and yellow in color. And, what's leaving the blood to go into the tubule is going to become urine that, includes those wastes and drugs and excess ions. We learned in that same lecture of blood vessels that we learned about reabsorption is the removal of substances from our blood. Now, solutes and fluid formed by our nephrons and collecting ducts and drain into the ureters as urine to be excreted out of the body. The rate of urinary excretion by any solute is equal to its rate of glomerular filtration plus its rate of secretion minus its rate of reabsorption. By filtering, reabsorbing, and secreting, our nephrons help maintain a homeostasis of our blood's volume, so how much blood we have, in its composition, meaning what's in the blood to go along with that water, all those ions and various solutes. Okay, let's talk more about filtration. Reading through glomerular filtration, we just said, is process in which water, gosh dang it, stinking screen keeps falling down. Is process in which water and some solutes in our blood plasma pass from the capillaries of our glomerulus across a 3-layer, what we call filtration membrane, which is what we're going to talk about here on the bottom right, into that capsular space, or Bowman's space as it's labeled as down here, and then eventually being pulled into our proximal convoluted tubule. That fluid that enters the capsular space is called glomerular filtrate, or I just call it filtrate a lot of times, and is just a precursor to urine. Okay. Together, glomerular capillaries and the podocytes, which completely encircle the capillaries, form this filtration membrane. So what we're looking at in picture B over here is just a very small segment somewhere, maybe we're just picking out right in here of that picture. And we show there are multiple layers to this filtration membrane, which has a sandwich-like assembly permitting filtration of water and small solutes, but not our big things in our blood, our plasma proteins, our red and white blood cells, and our platelets. And in order for us to pull stuff out of the blood, they must pass through the filtration membrane. So let's start at the interior, okay? Looking at the actual structure of the capillary, the glomerulus, looking at it, we see the wall of this glomerulus has a lot of holes in them. Okay, it's got fenestrations or pores. Okay, that are just think of like a strainer or a colander, if you call it that, where you dump your water and noodles after you boiled them when you're making spaghetti or mac and cheese. Okay, those holes or fenestrations are small, not big enough for our big blood cells and our platelets and proteins to leave the blood, but definitely easy enough for our small substances like our water, our ions, our glucose, and that kind of stuff. So the endothelium of the glomerular capillaries covered with large filtration pores, highly permeable to many substances, except red white blood cells. Therefore, glomerular filtrate has the same composition as our blood plasma, minus red blood cells, white blood cells, and our proteins. Presence of red blood cells or proteins in our urine would indicate that the pressure inside the glomerular capillaries is way too high, and we're just pushing them through the holes anyway. And there is an issue, or there's a defect in the glomerular membrane. There are little cells in between the capillaries called mesanglial cells that are also present between the afferent and efferent arteriole. And these are contractile cells that can also help regulate the formation of that glomerular filtrate. Directly outside of that epithelial layer is its basement membrane. Just like we saw with all capillaries, they have a layer of endothelial cells and a basement membrane. The basement membrane, shown here in blue, is very important. It's acellular, meaning it's not made up of any cells, but it's composed of minute collagen fibers and proteoglycans and glycoprotein matrix. A lot of biochemistry stuff in there. What's important, though, is that it has a negative charge. And a lot of the stuff in our blood has a negative charge. And earlier in the semester, we learned that opposites attract, but that like charges repel. So anything that's negatively charged, like our plasma proteins, aren't going to really want to go out into this area. They'd have to pass through this membrane to do so. Well, opposites or like charges repel. And so those plasma proteins are going to want to stay in the blood away from that negative basement membrane. Okay. Now those podocytes wrap around the outside of the capillaries, and they do so thanks to those leg-like or finger-like extensions called pedicels. But notice that those pedicels have little crevices in there. Now they're not completely empty spaces. They're covered with a membrane called a slit membrane. Okay. This slit membrane, or filtration slits as they're labeled as, are going to also carry a negative charge, providing another obstacle for large molecules. Okay. These are going to still allow very, very small substances. I'm talking super small, 0.006 to 0.007 microns or smaller. Still, that's a lot of stuff. Water, glucose, vitamins, amino acids, very small plasma proteins, ammonia, urea, and ions. So I always just think of, like having your capillary with two layers of like coffee filters surrounding it. And then you've got these little finger-like pedicels wrapping around that all together. So it just, creates a pretty big barrier to prevent the most important things in our blood, our red blood cells, our white blood cells, and our platelets, from leaving it and allowing all those little small things that I just mentioned out into the corpuscle space or the capsule space, where we then can filter out into the beginning of our renal tubule. So that's kind of the anatomical feature, the kind of smallest parts of a renal corpuscle. We are basically out of time. So we will start with this, and it's a perfect spot to, we've got the anatomy really kind of down. At this point, we'll pick up with glomerular filtration on Wednesday.